All right, very good afternoon, everyone. I hope with this particular clip, you got the context. We are talking about X-linked inheritance, some of the diseases like what we see in the royal family, hemophilia, and even otherwise color blindness. Uh, the molecular basis of these only came over the years of a lot of research, which led us to understand that what could be the reason at the molecular level, which is causing these kind of abnormalities. So I'm going to talk to you about chromosomal and molecular basis of inheritance. Especially, I'll just give the quick overview and recap of these concepts, which I've already uploaded previously, so that you are ahead prepared for this topic. Broadly, we are going to talk about some of the key experiments which were done at that time. We are discussing about genetics and genetic experiment. We discussed in the last class about Mendelian genetics. And now we'll be moving to the Drosophila-based Morgan's experiments, which also led us to establish some concept that how inheritance could also be sex-linked. Various type of concept of genetic recombination and linkage is involved in that. And then over the years, we also learned about chromosomal abnormalities, that how various type of aneuploidy and different type of syndromes may lead to abnormal babies. During the process of all this learning, we also had many other key biological experiments, molecular experiments, which led to establish that what is the transformation material, what is the genetic material which is transferred, is it the DNA or protein, how DNA replicates. Some of these are the very classical experiments which were done very elegantly at the time when there was not too much resources, not too much state-of-the-art instrumentation and technologies were available. So let's first start with Morgan's experiment. Here now what we are looking at, not the pea plant, but looking at this beautiful Drosophila melanogaster. Let me remind you that you are already seeing this Drosophila or the fruit fly when you have the banana peel somewhere, you throw it or lying in your kitchen. You see like those small flies, those are the fruit flies, Drosophila melanogaster. Of course, people don't do experiments like that. But for doing experiments in genetics, there's a quite a large number of genetic scientists who work on Drosophila melanogaster. Given that it is very small fly, very easy to cross, and also very easy to maintain. What you see on the right side are a lab view, which is having a small these kind of the tubes in which there is a media there, which is having this kind of nutrient like banana peel, and then these Drosophila are being propagated. They're grown over there. And it's very easy to culture them. It's very easy to maintain them. And it's very easy to, to have their generation, which is very fast. I just like to mention here, Dr. Pradeep Sinha. He's one of the key genetic scientists at IIT Kanpur. He worked very nicely on this model system to show various type of molecular understanding, how easy it is to do this experiment. So these are quite a bit of reality, which you will see happening across, and this is a very nice model system to study. But of course, what we are talking right now is the the elegant experiments done at the Columbia University by Thomas Hunt Morgan, who was trying to understand what Mendel did. Is that replicable, first of all? And if it is, then is there any association when you look at the complex organism, not from the plant, but from the insect and animals? Is there any association with the specific chromosome as well? Are there specific traits which Mendel was referring, those factors, are they also linked to any specific chromosome, not only just their passing and showing the dominant and recessive characteristics? So these are the some notation which I hope by now you are familiar with. If not, please do make sure that you are sitting with your notepad and your pen and trying to denote these things in your own hand so that you start getting more familiar. Now what we are looking at, female XX and male XY. This is the female chromosomes and the male chromosome, which you're aware of. Now what we are also looking at, a Drosophila fruit fly, which is having the red eye. That is the wild type feature. Wild type means that is commonly seen, most prevalent phenotype, which you will observe in all the fruit flies, which is essentially the red eye color. Now with this red eye colored female fruit fly wild type, now if you're crossing with a male, which is the XY, which is having the white color, where white is not so commonly found, and that is the mutant behavior. So anything which is not so common, that is a mutant. So what we are looking at now, multiple details here, XX female, XY male, and whenever we have the wild type feature in genetic experiment, they put a plus sign, right? Same gene donated for the wild type features with the plus sign and for the mutant without plus sign. This is a new notation which we are learning now. It's a good idea for you to start writing these things in your notepad. It will become easier for you to do these crosses. So wild type now, since both the chromosome on the X chromosomes, we have W plus W plus. What we see is the dominant phenotype or the wild type phenotype, which is the red colored eyes. Also, we have the X, small w and Y. So small w is the mutant phenotype. And that's why we see this particular fruit fly, which is male, is having the white eye color. Just imagine that if this was W plus, what should be the eye color? I'm sure your answer is it is going to be red colored if it is the W plus, right? So then it is not mutant, it's going to be also wild type. And here, since we are also trying to understand the location of the chromosome, it becomes important to pay attention to whether these genes are linked to X chromosome or Y chromosome. Of course, you will not know this thing. If there's any question comes, we will give you the hint that this property or trait is linked to which chromosome. So when Morgan is doing his experiment, he did the cross between wild type female fruit fly. So now this thing is what we will tell you in the questions, right? And you will write the notation yourself. Female fruit fly, XX, wild type means W plus, W plus, right? And a mutant white-eyed male. So male is XY and mutant is means for this property, it is small w. So what eye color will be the having the F1 and F2 offsprings. So like the Mendel's experiment, now this thing will follow the same pattern when we have the parental generation, where you can see the female red-eyed and the male white-eyed having the cross. All the offsprings, what is being generated here, are having the red eyes in the F1 generation. It means the red eye color is the dominant feature or the wild type most prevalent feature here. The same cross, if you continue now to the F1 to F2 generation. So now let's just try to make it more simplified for you to practice. Let's say when you are looking at this type of XY chromosome relation as well. So in this case, when we are saying female is having wild type eyes, so now let's say you put two chromosome, X chromosome, XX for the female, and on both you can denote this W plus as the feature which is going to define the eye color. Now likewise on the male, you have the Y chromosome and the X chromosome, but since the X chromosome is carrying this property, X is having the W, small w, and Y does not have that particular characteristic. So now if we take the gametes from the sperm and from the X or ova, X both are having the X chromosome having W plus property. So that's what you see here. X will having only one type of gamete, whereas the male will have both type of gametes, Y chromosome, which is seen here, and the X chromosome, which is carrying the mutant feature. So based on this, what you see, the female and male, which is formed here, both are having W plus, and W plus is sufficient to govern the wild type characteristic, which is dominant here, which is the red eye color. So all the first generation or the filial generation insect, which is born here, Drosophila fruit fly, they're all having the red eye color. But further, if we subsequently crossed, and just keep imagining what we have spoken for the purple versus white flower and the Mendelian experiments, same appears here as well so far, right? So now when you look at the gametes coming from the male as well as from the female, so now female is having this W plus and W both, and the male is carrying W plus and the Y chromosome, which is not having this characteristic. Now in the Punnett square, when you draw this, what you will see now, there are three still red colored and one is white eye colored. It means 3:1 ratio of phenotype is still maintained. So he was able to obtain the same type of conclusion what Mendel has done on the pea plants. But also here there is some more information here. Like for example, the female, one female is having the W plus on both the chromosomes, whereas one female is having only on one chromosome. Whereas the male, even on the one X chromosome, when it is having W plus, it is sufficient to give him the characteristic red eye color. Whereas when there is an absence of this W plus, it is going to give us the mutant feature, which is the white eye color. So what you can now gather from this experiment, that he was able to replicate Mendelian's inheritance and how the law of segregation works. Additionally, he could also provide some further insight that these genes must be located on the X or Y chromosome. And since this particular trait, which is linked to the eye color, is obtained or found on the X chromosome, that's why we see this type of segregation. So he said then there is a probability that these factors which Mendel has defined or units, they are indeed the Mendel's heritable factors, but they're actually located on some chromosomes, and there is a evidence based on these experiments. So very nice experiments done just on the fruit fly, looking at their insects which are born in the subsequent generation. They're able to come up with these conclusions. So based on these, and of course, as we go along with the more of the technology and now more of the next generation sequencing type of methodologies available, now we know that many of these sex-linked genes, they are located either on the X chromosome or on the Y chromosome. So the Y-linked genes, those which are on the Y chromosomes, they are quite few in number, which is around 78 to 80 genes, including the genes which help us to determine the male or female sex. And the X-linked genes, which are located only on the X chromosome, which is more than 1,000. And many of the characteristics, many of the diseases are actually linked to the female X chromosome. So now let's do a couple of crosses, which I think you should be ready on your notepad. Please do these experiments and these crosses yourself. You will really be very happy to see that any question which will come from this slide, which will be many actually, will be able to, you should be able to do that very easily if you can just follow these notations properly. So what we are looking at now, red-green color blindness. Like some people are not able to distinguish the differences between red and green color, and that is known as red-green color blindness. In fact, even in the army exams or in the railway exams, after doing all kind of scrutiny and final selection in the medical, they also check for the the red color or the green color blindness, because it becomes very critical when you are moving ahead with the looking at distinguishing these colors in the those minute decision making. So when these experiments which we are talking right now, there are three possibilities when we are looking at red-green color blindness. It means the female is normal and male is colorblind, or female is carrier and male is normal. Female is carrier and male is colorblind. How this detail came? Because here the gene which is responsible for the normal color vision, which is the capital N or the dominant allele, that is carried on the X chromosome, whereas the small n, which is the recessive allele, if there is a mutation over there, that is going to cause color blindness. So therefore, if you can just use these notations, what we have, unaffected individuals, which are just purely white, carriers, which are, let's say, the light pink color, and the colorblind individuals, which are the orange color. So now I'm going to give you this particular context. Please do this cross, and you'll be very happy that you can actually obtain a lot of information from these crosses. So what we are saying that female is normal. It means that, on the XX chromosome, when we have the capital N, that is the dominant allele, which is giving rise to normal color vision. Whereas in the male, if even on the, because it is having XY chromosome, even if the small n or the recessive allele is present on the X chromosome, that's going to make the male as a colorblind, because even with, it's not having two X chromosomes, it's only having one X chromosome. So even if the gene responsible for color blindness, if that is recessive in this case here, or having mutation, that is going to make this male as a color blind. So now if you do this cross here, so let's say the gametes obtained from the female, which is X capital N and X capital N. And now if you have the gametes from the sperm, which is X small n and the Y, you do this cross and the results are here, like X capital N, X small n, X capital N, X small n, and then in this case, X capital N and Y, X capital N and Y. So now what lot of information you can obtain from these experiments, the context which we are going to give you sometime, that, okay, if the in the offspring of these crosses or these, these parents, if the females are all carrier, what could be the, the gamete composition for male and female in the, let's say, parental generation, P generation. Or a colorblind father, like this is the father which is colorblind, is transmitting the only the allele to the mutant allele to the daughters only, right? So this is the right option because only the daughters having the XX chromosome are carrying this particular trait, which will be continued in the next generation. So a lot of these informations are there. Please take a minute and try to do this cross yourself. All right, so please do that. It's going to come to your quiz as well. In fact, I should not reveal it beforehand, but it will be coming at different stages, and a lot of questions will be asked just based on this slide and a lot of information which we have here. Very straightforward. All you have to understand these notations, right? So now the second context which comes here is that the female is a carrier and male is normal. So despite that both of them will appear normal, like if the marriage happens from this male and female, the female is also looking normal, but is actually having carrier, and male is normal. Now the gametes which come from it, X capital N and Y, and X are having X capital N, X small n. In this cross, since you know that even with one recessive allele here, which is small n, male will become colorblind because male is having XY, only one X chromosome. It does not have buffer like two X chromosomes like female. So in this case, male is going to become colorblind, despite that both the parents look normal. And that could be a question that, if one of the male born in the family is colorblind, despite their parents are both normal, what could be possibility? And in this case, the female is the carrier. And also we can conclude that there is a 50% chance that each of these daughters, the two daughters, two girls born, one will be, like her mother carrier, and other will be, normal, whereas 50% chance that the son is going to be colorblind or going to carry this disorder. So again, the terminologies may look difficult to you, but if you start doing these crosses yourself, you'll find everything is very straightforward here. It's very logical and quite easy. We will definitely, when we give you questions, we will give you help with some sort of gene notation or the clues of what is dominant, what is recessive. So do not worry about that part. Just worry if you can come up here and then can you do these crosses and can you come up with these conclusions. Now, third possibility, when it comes like this, when we have the female as a carrier and male as a colorblind. So in this case, of course, the male is already having the visible symptoms of being colorblind, but female is also not normal, is a carrier. And in this situation, one girl and one boy will be carrying this particular defect. They will be colorblind, and therefore multiple possibilities, multiple statements we can draw, like 50% chance that each child will have a disorder, like 50% in the family will have disorder. Daughters are having normal color vision, like the one which is having normal color vision will also be carrier in that context, and male will be totally normal. So many information you can obtain here. Just on the lighter note, I'd like to mention in the chat at sometime I saw long back in Telegram group that many students wanted to go for some wedding and some marriages, and time November, December is time, a lot of Indian weddings happens, right? So you must have gone and enjoyed those weddings, a lot of party and food, and of course music and dance. But what you'd have not noticed that in none of those marriages anybody would have done genetics test, right? So in India, this is like so much affluent weddings, lot of money being blown away for show off in these weddings, but the basic elements like doing genetic testing does not happen. That's not the concept, that does not come into our society. And I was just visiting one of the very small country, Tunisia, which you may not have heard name. And in that country, when I went for 2 years ago, that time for one of my collaborative research project, when we have joined India Tunisia project. So when I went to visit their lab, one of the key postdoc who was supposed to meet me, she was on leave on that day, which was very offending to me that I came all the way from India for this meeting and one person is missing today. And what I learned that, it was very urgent for her to go for the genetic counseling and genetic test on that day because she was going to get married next week. And today only she got the slot for the genetic test and genetic counseling. And it is considered so important over there that this kind of thing, they don't compromise. So before marriages, the genetic test, genetic crossing, genetic counseling, all these information has to be kept into consideration before marriages get finalized. And that is very impressive for me that so small countries, and they are so much knowledgeable and so much ahead in thinking about these kind of small details, which is so important. Like if you see here, many of these things could be avoided if you are already being tested beforehand, and also a lot of neonatal testing, like when child is at early stage, even during that time any test happens, all of those could decide that we don't see many abnormal childbirth and abnormalities happening. So this is just context to tell like why there is so much awareness required, and still we have to catch up on these small details. Nevertheless, Morgan was able to catch up on many details just based on these small fruit fly based experiments. He was not only paying attention to the Mendel's experiment and how they are following the trend in the Drosophila, but also he was looking at whether the genes what they being inherited, are they being exchanged in the recombination events during meiosis? Are there also some genes that are linked to the chromosome? So those kind of experiment he was trying to follow up. Now the next experiment which he did, he's looking at fruit fly, but more properties he's looking at. One is the wild type feature, which is the gray body color and the normal wings. Like wings are normal, which is the good size wing. And here, if you see, this wing is a small wing, which is the mutant phenotype. And then gray body is commonly seen. Again, that is the normal feature, whereas the black body is not so normally seen. That is the mutant feature. So if you look at the notation, you can draw it like B plus VG plus and dash on the another chromosome, B plus VG plus. So in this way, it will be easier for you, like B plus is the body plus, and VG is term for vestigial, but it is not vestigial, it is normal, so VG plus. So vestigial means like very minute or many very small, which is the notation used here. So now we have BVG and again dash BVG, which is the double mutant. So this is the notation which you have to at least learn in the Drosophila genetics, which is slightly different and slightly more complex than what you have learned in the Mendelian genetics. But this is giving us a lot of information which is not possible earlier. However, some key concepts remain the same. Like in this case also, the test, so in the F1 generation, what you see is still the wild type only appeared. The same feature what the wild type has is appearing. It means these are dominant characteristic and these are inherited in the first or the filial generation in the dihybrid cross. Now, if we're looking at further segregation of these, and then what we had discussed earlier in the Mendelian cross, like if you want to know the genotype, you can do test cross. And test cross was done with the recessive, right? If you recall. Same thing is here as well. If he wanted to do test cross, he did that using the homozygous recessive black body or vestigial wing male, which is the recessive characteristic, with this particular female, which is the dihybrid, having the normal features. So with this particular cross, now what you see like BVG or the small BVG, which is the recessive characteristic here for the body as well as the wing, vestigial wing, which is not having plus sign. Not having plus sign means it is a mutant form. Having the plus sign means it is a wild type feature. So in this particular, the male will have only the same gamete, BVG only, which is what you see in the sperm here for this cross. And X will have 4 possibilities of B plus VG plus, B plus VG, BVG plus, or BVG. Like so, both 4 possibilities are possible in this particular type of gametes which are generated from the egg. And based on this, when you do the cross, you can see these possibilities of having the wild type features, having the black normal feature, but also few flies generated which is having the black vestigial and gray vestigial feature, which led him to think that there are few genes which are probably present on the same chromosome, which are linked very closely to each other. Despite recombination events happening, there are genes which are also linked and they are crossing together. And that's how you can see some of these population emerging as well. So these are the very nice experiments which Morgan definitely added a lot more knowledge over what was known from the Mendel's experiment. Now, based on our more of the the genetic further updates, as well as the microscopy based more revolution happening. What we could now know a lot more about the chromosomes and the genes and different gene defects. And especially in these cases, what you see, like, if this is the normal chromosome, one of the segment of the chromosome is deleted just, during the various process happening of meiosis and further subsequent, in the fertilization process, embryo development. Sometimes these abnormalities do happen. And in this case, this is known as deletion. Means just a simple word deletion means something is being removed from the chromosome. So what you see here, A, B, C, E, F, G, H. Inversion, which is essentially now a segment of chromosome gets inverted. So it is like A, B, C, D, E became A, D, C, B, E, right? If you are given some of these examples, you need to know these terminologies. While we are not, so far, I hope you are sure that we are not asking you to mug around any specific terminologies, but these terminologies will be important for you to know. Deletion means you're deleting this fragment. Inversion, you are having one segment being inverted. And duplication, which means like, some part of chromosome is having two copies of that. So like A, B, C, D, E became A, B, C, B, C, D, which is duplicated. And then translocation means one part moved from one to other region, and that is a translocation, like A, B, C, D, E, and other chromosome is having MNO, PQR. Now this MNO gets replaced with AB, so now you got MNO, CDE, whereas AB came here, it became AB, PQR. This is known as translocation, and also more specifically known as reciprocal translocation, means both are reciprocating, both are moving from one to other chromosomes and getting exchanged, which is known as the reciprocal translocation. Whereas non-reciprocal translocation means only one is moving and other is not moving, which is happens less, and it is not shown in your book or in the figure. So these are the terminology which we have already described to you. And also, like many gene defects, many of these chromosome abnormalities are also seen in children. And sometimes these are actually things which are being seen in society. You will see sometimes this kind of appearance of the children. In this case, it is having Down syndrome, which is having aneuploidy. Some of the chromosomes becomes duplicated. Some of the chromosomes shows more copies. Sometimes some chromosomes are absent, or some chromosomes are getting deleted or inverted. These kind of changes happens, which led to the child with these defects. So Down syndrome, trisomy 21, means with the 21st chromosome having 3 copies, it should be ideally only 2 copies. Now it became 3 copies that led to the disease known as Down syndrome, based on the name of the scientist who discovered that. And the child looked like something like this. And, so now this is also known as trisomy for chromosome 21, means having the 1 chromosome 3 copies. Edward syndrome is same thing, is having the trisomy of the 18th chromosome, in this case for 21st chromosome. So now likewise, there are many other abnormalities, which Patau syndrome is having trisomy 13. You look at the hand abnormalities. Turner syndrome, when one of the copies of X chromosome is absent, and that led to this kind of feat. So many of these abnormalities are commonly seen in our nature. And again, if the genetic test being done beforehand or during the neonatal screening, a lot of these genetic tests could be done. Then many of these, although it could be a tough decision for the parent to decide, but still many of these things could be eliminated at the before the problem is actually seen. So now coming to some of the classical experiments of molecular basis of inheritance. One is about Griffith's experiment, which talks about transforming principle. What could be the transforming principle or factor? So he did one experiment to know that are there some genetic traits or genetic properties which is transferred from the different bacterial strain. And he used one bacteria, which is the Streptococcus pneumonia, having 2 types of characteristics. One is the smooth or the S strain, other is the R strain, rough strain. So S strain is the one which is having the capsule, and that one is actually causes the pneumonia disease. R strain or the rough strain does not cause pneumonia. So these are the 2 types of bacteria. And he was trying to do the experiment to know are there genetic traits which is transferred between the bacterial strain. For doing that, he did this experiment. When he first took, like he was trying to use this bacteria, inject in mouse, and trying to see what could be effect of this bacteria in mouse, looking at the pathogenicity of this particular bacteria. So when he used the S cell or the S strain, which is having the pneumonia causing ability because of the capsule which is present there, Of course, as expected, mouse died. When he took the R strain, which is not able to cause the pneumonia because it does not have the capsule. If you recall, when we talked about bacteria, prokaryote, eukaryote, we talked about capsule. So now this particular R cell does not have the pathogenic capability. Therefore, mouse remain healthy after the injection. When we have the S cell, which is heat killed, Streptococcus pneumonia, which is heat killed, it is a non-pathogenic. It becomes non-pathogenic because the virulence of that has gone, killed. It is now because of heat treatment, and therefore mouse remain healthy. Now, the last experiment, that was something which came unexpected. So he took the mixture of the S cells, which are the having capability to do virulence of the pathogenic characteristic, and the R cell, which are non-pathogenic. When he mixed the two, but here in this case, S cell was also heat killed, right? So it means this one and this one. He took these two and then tried to mix them together. And then when he is going to inject those, because it is heat killed, ideally one would expect it's not going to cause any pathogenicity. So as the R cell, which is also non-pathogenic, but because the two of them together is still able to have virulence, which is able to cause the mouse to die, then that led him to think there was some factor which got transformed because of the bacteria, which is having the S trait, which is the pathogenic trait, that bacteria having some substance, irritable substance, which somehow got transferred to the R cell. And because of that, the R cell, which was non-pathogenic, became pathogenic, and that led to the mouse to die. So this experiment was done by Griffith in 1928. The phenomenon is known as transformation or change in the genotype and phenotype because of the assimilation or integration of the external DNA by a cell. It means the DNA from the external S cell somehow came to the R cell, made it pathogenic, and as a result, mouse died. Another experiment which was done by the scientists Hershey and Chase, which was on the establishing, by now we know DNA is genetic material, but that time to test out what is the genetic material, is it protein or DNA, he used the phage, which is a virus, phage T2, and tested and did that experiment with the bacteria and virus, how they are going to, show the interaction, and how the phage, this is the virus, if it comes on the bacteria, what comes inside? Is that the DNA which comes inside or the protein which comes inside as the genetic material? So to do that, he used radioactive tracers where he labeled methionine protein component with the sulfur for the protein labeling, and the DNA was labeled with the phosphorus. So now if we look at two batches, in one batch, he used the 35 sulfur labeling, which labels for the protein. And in the second case, he labeled for the 32 phosphorus, which labels for DNA. And as you know, in the DNA backbone, we have the phosphate residues, and therefore that is going to label with the 32P. In proteins, we have the methionine, which is going to get labeled 35S. So as a result of it, all the proteins of the phage, which is the virus, is getting labeled either with the 35S or with the 32P. Now he wanted to know what comes inside here. Is that the blue thing, which is the 32P, or is that the red thing, which is 35S? For doing so, something like your mixer, he just mixed that. Now we have the centrifuges, which can do centrifugation at the high speed, so that the components can be separated. All the membrane and debris will come settle down in a tube, whereas the supernatant will come on top. And looking at those, that what comes into the supernatant, what comes into the pellet. Now in this experiment, in the pellet, he could see that the radioactive phage was found in the pellet, which led him to conclude that the DNA which was coming inside in the bacteria from the phage is actually, which is 32P labeled, is the genetic material which is coming there. So again, this is a very elegant and simple experiment which he proved using radioactive tracers. Third and the last experiment, I'm going to talk about the replication, which you have learned in the cell cycle phases, which we talked when we were discussing about the G1, S, G2, and the M phase, and how the DNA replication happens. So various scientists propose different hypotheses about dispersive theory, semi-conservative theory, conservative theory. Many of these theories were proposed to look at what could be the possible way of DNA to replicate. While there were different experiments done to prove and disprove all of them, finally the semi-conservative replication theory became popular based on the evidences which was provided by the scientists named Michelson and Stoll. what they did, and again, that time when we are talking about 18th century, a lot of experiments are happening. Those were mainly done with very basic setup, and radioactivity was one of the very popular way of lot of interrogation to look at the where the materials are going. So in this case, he grew the bacteria in the 15N or the heavy isotope media, which is shown in the blue color, dark blue color here. And when a small aliquot of that, the loop is being transferred to the another media, which is the 14N. So the another bacteria, which is bacterial batch, is growing in the 14N isotope. Now he transferred from here to there. Now when he did centrifugation, he could see in the after first generation or the first replication, something is settled at the middle. So again, when you look at ultracentrifuge, how the centrifugation happens, like the layers being formed after the based on the density of this particular content which is generated. So now this is the middle part, which is showing some density pattern, whereas subsequently in the second replication, he could see, in addition to the center, which is the very dense part, there is some small, less dense material also being seen in the top. So given that he knew that the 15N, which is heavy isotope labeled one, and the 14N, which is the lighter isotope labeled one, these are, that's how the bacteria are grown, and they are subsequently cultured. He could decipher, looking at the radioactivity tracing experiment, that the middle part was having hybrid band, which is having both heavy and the light bands, whereas the top one, which is this one, is having only light bands. So this is based on this, he proposed this theory that probably from the parent cell after the first replication, probably the one, the dark blue, which is coming from the parent, remains consistent in both of these DNA, which is being replicated, and the new material, which is the light blue, is being generated, which is now making a double-stranded DNA. And subsequently, in the next generation, now we have the dark and the light blue, but also from the light blue, we got both the light blue made, because from the dark one, you got another light blue synthesized. And therefore, in the second generation, now you got the hybrid of these two, whereas also you got the 50% of the light, which is the 14N lighter isotope. So based on these experiment and providing the evidence based on this experiment, Mendelssohn and Stahl's theory of DNA replication, which is semi-conservative, became popular and became acceptable. So just to summarize, I think we tried to give you, and of course, you should have watched this lecture, but again, probably things will be much clearer to you, that we talked about some of the elegant Morgan's experiment, sex-linked inheritance, which Morgan proved that there is a basis of where the genes are located on the chromosomes, some of the key events happening for the genetic recombination, linkage, and various chromosomal abnormality, which we now know based on the biochemical and genetic testing. I also try to re-emphasize and give you a message to do these testing. Whenever you see in society things are not happening, do educate and do provide more awareness for these type of activities. And of course, Griffith, Hershey, Chase, Mendelssohn, Stahl, some of these are classical experiment, but very nice conclusions were drawn, very fundamental knowledge was generated from those experiments, which is still remains valid despite doing the experiment on a very limited resources at that time. So I hope these experiment, these knowledge obtained for the chromosomal basis of inheritance and molecular basis of inheritance seems interesting to you, and you will definitely find these things quite logical. Where if you do these crosses, these experiments yourself on your sheet, then you will find that there is a very much logic involved in doing these things, and there is not too much mugging around to know these factors, but rather you can follow a pattern and trend if you can do these things yourself. Now, last 5 to 10 minutes or so, I'll just quickly talk a little bit about our own work. During the entire 7 lectures, this is today being the last lecture. Of course, we did not try to find time to talk about what we do at IIT Bombay, but just to get you some idea, like my lab works in the area of proteomics, which is, we have only talked about gene technologies and genomics, but what is coming more recent is the from genomics moving to the proteomics, which means looking at the protein at the high throughput manner and trying to understand many diseases by looking at the entire proteome or all the proteins. Your textbook had a couple of examples, but not too much that I thought I should take in this course. But I just thought to quickly show you a couple of things here. While I was getting trained in the area of proteomics during my PhD and postdoc, we're also doing a couple of workshop and training programs. Now, this experiments were done actually during a training which I was conducting at the New York and Cold Spring Harbor. Imagine the beautiful location like this when you can do these courses and conduct these training programs. But what was very important that the experiments has to be done with great precision, and all these steps which we perform in the doing these type of proteomics experiments has to be very much timed with different steps involved. And I did not know somebody was recording during the while I was conducting this course, and so many timers are there for each step and every reagent which has to be put in for doing these experiment to look for what is happening in the healthy versus cancer and different type of protein changes. So it also emphasized finally that okay, the experiment got failed because one person made some mistake, and that means it's also a lot of team spirit which is involved. So doing experiments are not only just the doing your own individual work, but also it brings lot of team effort. If anybody makes any errors, any mistake, that becomes very difficult for others to pinpoint, and everybody has to be very accurate. So therefore, learning science and research and doing some of these experience will probably be enriching for you as you go along in your career. It will be good to get some exposure in different labs which are having some of the research training capabilities. My lab works essentially in the broad area of proteomics and system biology to look at how to do various translation for different diseases. And while we are quite keen in developing some of the technology platforms and new test and assay development, but primarily lab looks into two major biology projects or clinical projects on the infectious disease, especially malaria and now COVID-19, and also on the cancer, especially brain tumors. And then given that we are having the National Proteomics Facility, many other model organisms from collaborators are also being studied. So we have developed many technologies which could look at the proteins in the separating based on their properties, and those are known as two-dimensional electrophoresis or various type of gel-based proteomics platforms. Some of the mass spectrometry-based approaches like multi-TOF can actually detect what could be the mass of these proteins and peptides and how to separate these complex mixture. If in a given tube you have been given 10,000 protein or 5,000 protein, imagine that now we have instruments and technologies which could actually segregate all of that, and we can understand what are all those proteins there and what is their quantification. Also, we can look at the proteins in the genome-wide scale, all the thousands of protein at the protein microarray level, how proteins can be tested with the drug interaction, and how protein can also interact with protein or other biomolecule can be tested with technologies like surface plasmon resonance. Throughout, I have put the key people who have been involved in the lab, the PhD students or postdocs who have developed in these technologies and who have been keenly involved in these projects. So to acknowledge them, I have put over there. Then based on all our work, we were funded very generously from Government of India to establish the Proteomics National Facility, which is quite equipped with various type of mass spectrometers. Mass spectrometer, the simple idea is to measure accurately the mass. If you look at all the Olympic games, when the athlete being tested, when they take any kind of drugs or any other thing to be tested out before the game starts, all these being tested with the mass spectrometer. Different type of mass spectrometers are there to look at all the proteins, all the small molecules, drugs, or looking at the specific test for selected peptides. So different type of configurations of mass specs are there, which is generously supported by Government of India, where we also utilize these mass spectrometer, how to develop the clinical test using these technologies and looking at a specific protein-based test. And again, some of these tests are quite close for the clinical testing. Now, just briefly, I'll talk about some projects on the infectious diseases like malaria, proteomics, and COVID-19 more recently. So for malarial project, we had collaboration from many clinicians and doctors from different endemic part of India who are from different parts, especially from the Maharashtra, Mumbai to Orissa to Goa, even Rajasthan, Bikaner, and Kolkata, different parts of India, to look at the some endemic regions of malaria. And what we have found more interesting over the years, that now there are two different organisms which causes malaria. One is falciparum, one is vivax. But vivax is more on the rise. Almost a decade ago, 10 years ago, falciparum was mainly causing malaria, but now more of the vivax is causing malaria. But our understanding of vivax malaria is not much. So what are the possible biomarkers for vivax malaria? That is something is still not very clearly known. And therefore, we looked at various type of patient samples, like their blood sample and the parasite samples, to find out what could be the specific peptides or proteins which is only present in vivax and not present in the falciparum. And looking at those, we can identify now some novel candidates which could be taken forward for the developing the YVAX specific test. Further, malaria causes different type of severe form of malaria as well. And there is no clear understanding how the same malaria can actually cause different type of severity, like severe falciparum, cerebral malaria, severe anemia. Different type of complication happens as a result of the malaria pathogenesis. So we showed in this paper published last year, Nature Communication Biology, how the same malaria from the mild form becomes severe form, and what is the unique signatures and unique mechanism which can lead to these type of disease pathogenesis. And again, I'm not going to talk in great detail about the these biology part of it, but just giving you a flavor how these technologies can actually dissect out and provide us more valuable information like these. So because we had many collaboration from many doctors who are working on different parts of the India for the infectious diseases, when the COVID-19 emerged, the same set of clinicians, especially Dr. Shastri from Kasturba Hospital, she's head over there, Dr. Om Srivastav from the Jaslok Hospital director over there, and many other collaborators, we thought to investigate the COVID-19 problem, and getting the sample and all those things were very much easy for us because we already had these ongoing projects with these clinicians. But doing the project was, of course, very challenging. As you can see, all the lab is totally with the PPE and all kind of biosafety considerations to be followed. But it was, of course, most challenging but very interesting set of experiments which we performed during this time. An idea was from the nasopharyngeal swab sample, can we look at all the protein which are present over there from the human side and from the viral side, because all the nasal route will have the host protein as well as the viral COVID-19 SARS-CoV-2 infection. If there is there, some of the viral protein will be there. So the CT values which we obtained for each patient from the doctors with RT-PCR, the same sample were tested with our mass spectrometer, and then we could actually find many peptides which are unique for the SARS-CoV-2, like spike protein, nucleoprotein, and replicase polyproteins. And we then try to develop the selected reaction monitoring, or the SRM test, looking at these specific peptides of virus. Further, what was not known from any technologies and what is not possible from any other technology which we could do using proteomics is how a patient is different in the mild, mild form of the COVID-19 and severe form of COVID-19. Imagine that time when we had second wave and huge number of population was getting infected. Everybody who gets COVID-19 becomes panic that whether they're going to get severe or they're going to remain mild. And there were no sufficient ICU and the infrastructure available that everybody could be given priority for the severe treatment. So we thought to look at almost in 200 or so patients blood sample which is either negative or the mild form of COVID-19 or the severe form of COVID-19, try to look at their blood, remove some of the abundant protein, look at all the protein present in the human blood. In response to the viral infection, what happens to human host? And then looking at various type of technological testing, we could come up with this test, which is based on the mass spectrometer, which indicated that the patients, if we look at the proteins like angiotensinogen, apolipo-B protein, fibrinogen gamma chain protein, serpin G1 protein, the levels are very different in the mild and the severe form of disease. And these could be early indicator to tell that these individuals might be getting prone for the severe form of disease. So again, some of these things became very novel, and we patented that, and a lot of highlights in different journals we had for this type of work. But another major project in the lab is on the cancer, which is with the Tata Memorial Hospital and ACTRAC in Mumbai, which is Asia's largest hospital, where we look at many deadly brain tumors like meningiomas, gliomas, and medulloblastoma, using our technologies, which we have been talking briefly here, taking the patient sample. We directly work with these doctors and directly work on the patient samples of the brain tumor tissues and the blood samples or the cerebrospinal fluid samples. Look at using our technologies, what kind of changes are happening in the brain tumor patient as compared to the normal controls. Again, using these type of proteomics-based investigations, what we could see, like there's a difference into control versus grade 1, control versus the grade 2s. And when grade 1s and grade 2s are quite different, even they may be forming some different subtypes. And therefore, when we look at more closely here, these are the two different type of controls. These are grade 1 patient and these are grade 2 patients. So the grade 1 patients are forming two cohorts or two subtypes. It means not all early grade of the brain tumor patients are same, and they should be given different treatment depending on their location of the tumor and their various other features, which we can understand. Also, can we look at the the cancer detection early, and that is one of the very challenging question for clinical deployment. When we go for our normal blood test for the routine health checkup, is it possible to get some ideas, some clues to do early test for the cancer? And that's what we talked in the earlier context as well. In this case, we had these 200 plus brain tumor patients of grade 2, grade 3, and grade 4 of gliomas. And looking at the entire pattern, we could only find 4 proteins, which is showing the pattern from the low grade to the high grade. And these proteins, sortin nexin 1 and some of the IGSG1 proteins, they show a very nice pattern that very early itself you can pick up the disease progression, and they could be good protein for testing for the reduction of the tumor. Also, these technologies can give us a lot of clinical insight about how a patient is going to perform. Is that going to be more aggressive type or less aggressive type? Looking at these, we developed a test based on the NED9 protein, which could differentiate that tumor. They're both tumor patient, but one is more aggressive, one is less aggressive. Can we find out some clues based on that? Can we find some clues based on the mutation, which is happening in some genes like IDH, associated dehydrogenase, which is important gene for the cancer mutation in this case. We also look at the pediatric brain tumors, which is the children which are having the brain tumors. And so far, it's not very clear why these two type of brain tumor, one is very aggressive and one is not so aggressive. And again, I would like to mention that some of the students like you who are pursuing the engineering also sometimes work with us on different projects. And one of them was Anurag, who was from computer science department. He did very nice work of looking at machine learning algorithm to find out the best possible clinical markers which could distinguish group 3 and group 4 tumors. And based on his identified markers, we obtained data from Broad Institute. They are also working on the medulloblastoma. And this test, what is shown is the independent test done on the Caucasian population from US data set, which Anurag was able to very efficiently distinguish which patients are group 4 and which are group 3 and which are group 4 only with one misclassifier, which was very promising because this was independently done without knowing that which ones are what. And we just got the clinical data from those patients. And we had our own gene signature based on these 6 protein gene signature. He was able to distinguish them well. And now we have some better understanding about why this disease is very aggressive and where we can do some of the clinical experiment to try to control these diseases. So these are the areas which we work on. And of course, while doing so, a lot of big data being generated. The brain research is of course very fascinating, but there is less resource available. Still a lot of data is quite missing in this area. While working on different brain tumor, we realized that the data even for the normal brain proteins are not present. We do not know how to compare with different brain regions. So in that context, we had some collaboration from a Spanish team when we have now developed the human brain proteome map, which was published last month in the cover page of Journal of Proteome Research. And we have also made a BrainProt, a public repository available, which is able to provide a lot of information for the community now. And finally, I'd like to highlight that some of the training program which we conduct in this area, some of our own research work in the cutting edge of proteomics is all available on various videos and various details which we have in this Proteomics IT Bombay YouTube. If any one of you have interest, you can definitely follow some more details over there. Finally, let me just show this clip and then I will take some more questions from all of you.
We are proud to announce the high-resolution mass spectrometry-based proteomics research and training facility set up at IIT Bombay. PPP-funded high-resolution mass spectrometry facility will cater to the increasing needs of the Indian research community. With the help of state-of-the-art proteomics instrumentations, the facility aims at offering services ranging from proteomic and metabolomic investigations of samples, understanding PTMs in complex samples, validating of target proteins and metabolites, and their relative and absolute quantitation. The community can also benefit from access to other platforms available at IIT Bombay, like microarray, Maltitop, and surface plasmon resonance, all under the same roof. We also intend to offer high-quality training and educational programs in proteomics. We strongly believe in transgenerational capacity to advance the proteomics knowledge for the benefits of global science and societal development.
Come join us to be a part of one of the largest proteomics facility in India to realize the dream of late President Dr. Kalam and make India a superpower in proteomics and big data science.
So I'm just going to review the content what we have covered so far in this course. BB101 MCB module, and also some of the guidelines for my section of MCB for the midsem. So I think, just to remind you, we have been discussing about from the first lecture, various cell organelle, different type of prokaryotic and eukaryotic cell. And of course, we try to emphasize in this particular part that how it is important for us to introduce all of you who are engineering aspirant at early career stage to learn about some of these basic knowledge, which eventually may help you at some point to come up with some breakthrough ideas. Now, the second lecture was essentially dealing more with the cell division and cell cycle, where we tried to show you different interplay of cyclins and cyclin-dependent kinases, how different type of cell division happens from mitosis versus meiosis. And during the process, you learned also some of the basics of the cell organelle. Moving on to the next lecture, when we continued with the cell cycle and DNA tools, we introduced you to the ideas of gene cloning and recombinant DNA technology. Also, I showed you this clip, which will probably remain with you to be very ethical and not to perform any misconduct in your career, which was apparent from these cloning experiments which we have discussed. Now, moving to the genomics and DNA tool, that was a lecture which we talked for the new technologies, especially polymerase chain reaction, how different gene expression technologies, including some of the next generation sequencing and microarray based approaches, can be used to understand the diseases and organism better. So those knowledge which we obtain from various biotechnology tools, which is quite apparent in this type of pandemic time, when you hear that we found the new mutant, new variants, all those are only possible to learn based on these technologies, what we are talking in this course. Further, there was a session based on the bioinformatics, which we try to give you some idea that how you can use some of these tools yourself, what we are talking, and try to gather some information for the sequence alignments and looking at different type of phylogenetic trees. I also uploaded one lecture on the metabolism for your better understanding about the photosynthesis and respiration. And we showed you some of the hands-on activity for which you did a quiz and assignment based on the bioinformatic analysis. Moving on, we then focused on last two lectures about genetics. Last lecture was essentially about Mendelian genetics. I also gave you the video clip directly from the class, which would have at least made you feel that you're sitting in the class of IIT Bombay, and learned about the Mendelian rules of law of segregation and monohybrid and dihybrid crosses, right? And finally, we have reached today when we talked about chromosomal and molecular basis of inheritance, and this clip is again reminding that how these gene defects do occur, and one could understand them better now with these kind of DNA tools which we have been discussing in this course. So all of these are the part of the 7 lecture which I have talked in this particular module so far, and this is a summary of what how we are going to take it forward. From the 7 lecture which we have given, which I have delivered so far, the 5th lecture, which was the bioinformatics, some hands-on activity, and the metabolism lecture, that I decided not to use for your midsem. So that will be a bit of relief for you to not go through those details once more and not to memorize many of the enzyme name and pathways. But for your own understanding, it's really important to learn from those lectures and do try to understand those concepts. But not everything is required for exam. So I just thought to drop that one based on some of your requests. Now, so therefore, in the midsem from my part will be 6 lectures, which will be 1, 2, 3, 4, 6, and 7. Now, please don't ask these questions again and again later on in the email or other Moodle forum that whether metabolism will come or not, bioinformatics will come or not, or this will come or that will come. That's why I thought to talk to you and give you a very clear idea what is going to be covered. Additionally, we are going to use only MCQ, which will be multiple choice questions for the 20 marks from my part. So 25 marks will be coming from my section of MCB, where 5 marks we are going to use from the quizzes which you have done so far, where we are going to take the best 5 quizzes out of 8. 7 have been done so far today. And then one more is going to be conducted in the tutorial session shortly, right? So the tutorial was again the challenge of the T4, the 4th tutorial when to conduct, because you have the quiz in that slot. So looking at the poll and looking at the logistics that just before Christmas and just on the day of Christmas, it's not good to organize a session where most of you would like to relax and use your time with your family. And a lot of people actually opine for the Wednesday evening slot. So we are going to have the next tutorial on the 22nd December, 7 to 8 p.m., where you will have a quiz, which will be 8th quiz, based on these two lectures together, 6th and 7th lecture together. It is going to be a good refresher for you to understand these concepts once more and be ready, because these are going to come back to you again in your midsem, right? And let me introduce you to next instructor. Professor Shilaja Nair is going to be your next instructor for the MCB module. She is going to now take the next 7 lectures from now onwards, from Thursday till the just before the midsem. She will further build your knowledge into various type of fundamental concepts and applications, which we would like to emphasize in the molecular and cell biology module. Finally, I'd like to just remind you that, in the course we tried many of these experiments to show you, right?
Take 3 separate drops of
These
blood.
experiments will definitely not
Place
come
a
in the
cotton
exam,
rod on the finger to stop the blood vessels.
but just to make sure that you are also,
as a DP and monoclonal D on
despite
each
not
drop of
able to
blood
come to the lab,
respectively.
you are still able to appreciate how biological experiments and conclusions being drawn. So you can, aim was to show you many experiments live from the lab and show you how the cell looks like, how gram staining performed, how PCR being done, how blood grouping done, to really give you a flavor of the logic and technologies involved in doing biology and bioresearch and clinical research. But of course, not those that are going to be used for your actual testing in your exam. So nevertheless, knowledge associated with that is definitely valuable for you to utilize in your question, which we have been asking you in the different quizzes. Uh, Finally, I'd like to just recap and remind you that we have given you many examples, which is like food for thoughts, where we try to give you more futuristic vision how biology is really transforming and going to further transform the whole humanity. In which way we are looking at precision medicine and personalized medicine. It's going to be very important to look at diseases in much more detail, especially like cancer. We gave you example of Angelina Jolie and Lucas Wortman, how different type of tests being used to detect the circulating tumor cells. And if you have watched the metabolism lecture, one of the clinician, Dr. Ali, talking about the brain tumors and diagnosis with the new technologies. So again, all of these were just an attempt to really break the barrier that, you should not feel that this is only for biologists and biology and clinicians, and this is only for engineers. You will see an interplay, how much technologies are required to really learn from each other, right? All right, so this is where I'm going to end the course. Right, so key here is from this, the theory of everything, that there is always hope, and we should break the boundary. We should think about how to really strive in achieving the human endeavors, where there should not be any boundary. When there is a life, there is hope, from the Stephen Hawkins. And of course, during the course, we have tried to break that boundary, tried to break that barrier of being biologists or being engineers. We talked about the bigger things, bigger pictures, where all of us fits into the puzzle with the small pieces, and together, how well we can try to solve any of the complex problems. I hope in the process you have also learned the need to be more ethical and show your high quality originality in your research and publications. Also, the Darwinism, the evolutionary theories of only the best who are adapting will be surviving, also remains pretty much valid, especially if you see, like, how quickly you are able to adapt some of the biological concept and started doing so well here. That's pretty much it from my side for BB101 MCB module. Good luck for the entire course and to your journey to IT Bombay. And that's pretty much, I'd like to stop it here. If in case you have some questions and queries, I'll be happy to take it forward.